Thank you so much for joining us, Mark. I know you're going to join us in person, but we all have to listen to our doctor from time to time. So I appreciate that you are still available to talk despite the hurdle. If you could just briefly introduce yourself and share a bit about the pre-symptomatic framework for your patient activism.
Sure. Yeah, my name is Mark Harrington. I'm the Executive Director of Treatment Action Group. We were formed in 1992 by a group of activists from ACT UP New York who were members of its Treatment and Data Committee, and we had been working since early 1998 on 1988 on. We started out by working on trying to get the Food and Drug Administration to change the way that they oversaw and regulated clinical trials for people with HIV and AIDS. And then we got more involved with all aspects of the research, including drug company sponsored research, National Institutes of Health sponsored research, and FDA regulation. And in thinking about this panel and our talks that we've had previously, a couple things came to my mind about, some similarities to, or at least lessons learned from HIV activism, particularly in the early years, because back in the 80s, AIDS was a new disease. It was first notified by the Centers for Disease Control in 1981, and the virus wasn't discovered until 1983. And throughout the 80s, there was a lot of uncertainty about the meaning of an HIV diagnosis for a otherwise healthy individual. Most of the people that had come into being infected with HIV were young and were a very diverse group of people, including young gay men, bisexual men, women who had sex with a man with HIV or had shared a needle in terms of injecting drug use with somebody with HIV. People that could get, it was bloodborne, it's bloodborne, so you can also get it directly from sharing a needle with an infected person. You could also get it from a blood transfusion or from factor products that were used by people with hemophilia. You could get it from vertical transmission in utero or postpartum from your mother, or as a nursing infant from your mother. And then there was some individuals from different countries that it seemed pretty obvious by 1984 that HIV was present in Haiti, and it was present in what was called Zaire, which is now known as the Democratic Republic of the Congo. So there was a lot of hints that the disease AIDS might be caused by this virus, but it wasn't really definitively shown until 83. And throughout the 80s, I would say that it wasn't really clear what the prognosis was for people that had HIV that were otherwise healthy, because most of them had not progressed to full-blown AIDS, and AIDS itself is a constellation of different conditions that are all associated with HIV infection, and it turned out they were caused by HIV infection, which HIV attacks the immune system and wipes out a key group of cells, the CD4 T cells, that coordinate an effective immune response against a number of pathogens, viruses, bacteria, fungi. And it was also associated with a bunch of cancers, also with dementia and with wasting syndrome. So in the late 80s, when we were in ACT UP, there was a lot of discussion about what did an HIV positive diagnosis mean? Did it mean that that you were going to get AIDS, or did it mean that you might, some people might and some people might not? And it turned out that by the late 80s, it became clear that almost everybody that was infected with HIV was indeed going to progress to AIDS. And that raised the issue of whether or not studies should be done with drugs that were being developed to stop HIV. Should they be given earlier in the course of progression? Because the first drug that was approved by the FDA was AZT. It was approved in early 1987 for the treatment of AIDS, but only of people that had a full-blown AIDS and a CD4 T cell count of below 200 per droplet of blood. And really the next more than 20 years were spent on not only trying to find effective combination therapy because the virus mutates a lot. And we had, by 1996, we had effective 3-drug combination therapy that could stop HIV from replicating, prevent the development of resistance, and either protect the immune system or restore the immune system almost to as well as it was before infection. And then it wasn't until 2015 that research showed that actually anybody with HIV should start triple therapy immediately after finding out they were infected, and that would have an impact on their survival, and not only on their survival, but on reducing their risk of transmitting HIV to others. And so those clinical trials only happened because they were pushed for by activists like people from TAG and ACT UP and other activist organizations, because we wanted to know, and we also knew that health organizations wouldn't be pushing treatment for everyone unless there was really strong scientific evidence that it worked. And so it took, more than 3 decades for effective treatment. But in those years, we had also, brokered a lot of, and many times demonstrations were part of the brokering, but we had brokered effective partnerships with people at the FDA, people at the NIH, and people in pharma, and with our own doctors, and created a kind of a movement that's pretty unusual, where patients are central to research, design and implementation, guidelines design and implementation, and regulatory design and implementation. And so I think those are all areas where people with a genetic propensity to ALS could, benefit from some of those practices. And I would want to say first, before I should have said this really at the beginning, but just by being an activist and by being mobilized together to work to help this problem, you're already making a difference, and you're already creating science-based and evidence-based hope that progress will be made and can and should be made. And you also have a right for science to be done on your condition, and you have a right to participate in clinical trials, and you have a right to demand that regulators and researchers study the condition as it is now and before you get clinical ALS. And so I'm very inspired by what you and your allies are doing, and fully supportive.
Well, thank you so much. That is very kind words, and the example that you guys have set, of course, inspired so many people, and you changed how the FDA handles like everything, not just for HIV patients, because they, at least on paper and in voice, will say that they're incorporating patient perspectives and everything, and I have found them to be pretty open and pretty collaborative with us. So thank you for that, for changing the game for all patient advocates. If you could, was there ever pushback on the tension between the community of wanting to address the problem early, or even keep track of the problem, monitor the problem, versus people saying, well, we got to worry about these people with full-blown disease, and you're just taking away from the people suffering from the full phenotype. So just curious if that was part of the dialogue at that time, or not so much.
Well, there was a tension at the time because there's a different risk benefit for research when it's done on people that are asymptomatic or otherwise healthy than when they're already sick. And on the other hand, because HIV by 83 was determined to be an infectious disease, and indeed an epidemic and a pandemic, and because there's precedent with other infections for earlier treatment being needed. One example is tuberculosis, where TB treatment started in the 40s, and the 50s was already being treated with 3 drugs together. And then there's an earlier form of TB called latent TB that it will develop or could develop into full-blown disease, but not in everyone, unlike HIV. Well, I should point out that even with HIV, there's a group of people that have genetic factors that make them slow progressors, and some of them actually don't need to go on to HIV treatment for years or even decades. That's less than 1%, but it's still an important biological fact. But the debates were more about, they became more about monitoring risk levels by biomarkers, and a whole slew of biomarkers were evaluated, but most of the clinical trials eventually focused on either the CD4 T cell levels, which is the tissue that's attacked by HIV and goes away, is actually destroyed when you get AIDS, or HIV viral load levels in the blood. And the PCR test for that came about in the early 90s and revolutionized the space. And then viral load became a surrogate marker that was approved by the FDA for use in all HIV drug trials in the mid-90s. And so none of the drugs now are subjected to full clinical endpoint testing, and so that you can get a drug fully approved based on 48 to 96 weeks of changes in the viral load, which is a sustained reduction to undetectable. So I believe that my understanding is not full or great about ALS, but my understanding is that there's not an agreed biomarker or set of biomarkers that clearly predict how quickly somebody will get sick. I may be wrong about that. And I don't even know about the genetic mutations that predict or predispose people to getting ALS, or whether they like are 100% predictive, or whether they only work for some people. But in any case, none of those reasons are good reasons to not conduct research in space if there's good scientific rationale. And I know that the space has also been experiencing some setbacks recently with drugs that had received accelerated approval that ended up not working. And the sponsors actually did the right thing, which was to remove them from the market. But it's those kind of setbacks are common in research early in diseases. And while terribly disappointing, it's actually good to find out that things don't work, because you can invest scientific resources, but also your own resources in better, more promising approaches.
Yes, excellent analysis. So wonderful to hear about, the biomarker trajectory in HIV. And just to share, without getting into the specifics, there was that first era of the T cell, CD4 T cells that were a byproduct of the disease, and thus imperfect because it's not the disease itself. And then you discovered this viral load, which is the disease itself. And, I think that's always the perfect biomarker, if you can measure the actual, what is doing the actual damage to the body and not a byproduct of it. And I think we're just getting to, there was one drug approval for one form of genetic ALS based on a non-specific, downstream marker of disease. And we're just in a time period where it's like, how applicable is that to everyone? And where could it be used? And, I think, we're just hopeful that even if it's not the perfect disease itself biomarker, that it could be used for smarter, shorter, better trials, and including more people at different disease stages.
Yeah.
But anyway.
I mean, I think it's also important to recognize that medications that resolve symptoms, even if they don't resolve the underlying cause of the disease, could also be candidates for approval and for use clinically. And one of the paradoxes that will come up in that case is that if they provide symptomatic relief, would they also have some effect in delaying the progression of the disease? And I think that would have to be decided on a case-by-case basis. But I think there's a great hunger to simplify sometimes that ends up being premature. So for example, the discovery of highly effective HIV treatment did not take away the need for prevention or treatment of opportunistic infections when they occur. That still is needed. However, it greatly reduced their occurrence.
Well, wonderful. Well, I think I'll just bring up one more thing. I was just thinking about it, and there might be parallels in our patient movements. When you were listing the populations who are most at risk of acquiring HIV, at least in the United States, and if stigma from the medical field would seep into the research, or and how you guys dealt with stigma. Just to share briefly,
Mhm.
there's not a moralistic stigma necessarily associated with us and projected onto us by the researchers who are studying us, but there is sort of very comfortable statements of categorical um, denigration of behavior, lifelong cognitive and behavioral issues, perhaps, that are pretty subjective and speculative, but are pretty stigmatizing and demeaning. And we're just trying to figure out how to handle that in discussion with scientists. And I wonder if that came up in the HIV patient movement, and how you guys handled that.
Well, stigma has been omnipresent in the HIV movement since the beginning, because the social environment during the Reagan administration was highly homophobic. It was highly misogynistic, and it was highly racist, and it was highly stigmatizing of people that had a history of using or were still using injecting drugs. And so almost every group, and then because those groups were stigmatized, anyone else that got HIV or AIDS was almost like presumably stigmatized. So there was a huge, and there still is a huge stigma about HIV. But I think the other part of it, which is less, maybe less moralistic, is just the horrifying nature of AIDS as a disease, and its lifelong nature, the fact that it can't be cured. And people were judgmental about the behaviors that could lead to people becoming infected, or then they could turn it into stigmatizing of people that are not taking their medications, which sometimes may be out of an individual's control. If there's a stock out of drugs, if there's an insurance problem, if the person doesn't have insurance and can't get onto an assistance program, there's all sorts of reasons why, and barriers that could, if you have a lot of stigma or shame, could make things even harder. And a lot of people that still die of AIDS now have wrestled with stigma, and stigma has could make their outcomes worse. And it also can create situations where researchers or physicians, this happens a lot in Hep C, which we also work in, they don't think their patients are, committed to treatment or make rational decisions, because they may still be injecting drugs. And so there's even a kind of a notion of a deserving versus an undeserving patient. And that occurred with AIDS as well, where people would talk about innocent victims, and innocent victims would be like pregnant women or children, and then everybody else, I guess, would be presumably not like a guilty victim. Whereas, of course, in reality, everyone's innocent, and even the use of the word victim was problematic, and we tried to get rid of it in the HIV space as far back as '83. But yeah, stigma can be really bad, and it can also, I think by its nature, it can sometimes manifest itself in people who mean well but aren't thinking through how they may come off.
Well, thank you so much for breaking that down, and we'll try to keep an eye on that, because we certainly don't want to be in a position where stigma is keeping people from seeking care when there is care, or treatment when there is treatment. And
Yeah.
thank you so much for this talk.
Sure.
And I
Hope to see you
appreciate
guys later.
yeah, hopefully later too. Thank
Okay.
you so much, Mark. I really appreciate it. Bye.
Okay.